So welcome to the Precision Medicine in IO panel at the SACS conference. I'm Bob Silverman. I'm the Chief Business Officer for Alloy Therapeutics, and I'll be moderating this panel. Prior to Alloy Therapeutics, which is a startup company in the biotech space, I was with Roche for a long time, business developer, many different roles and responsibilities, including in the diagnostics arena of Roche, which is why I was, I believe, asked to moderate this panel. We have with us today an esteemed group of panelists that range across the entire ecosystem and value chain of drug discovery, drug development, representation from academia, from industry, and industry includes both pharmaceutical and diagnostic industry. We have leaders of consortium, we have regulatory expertise on this panel. And so what we're gonna do is we're gonna take a journey across the pharmaceutical value chain. We're gonna talk about how to consider precision medicine applies to immuno-oncology from the perspective of decreasing clinical trial and error of medicine. Increasing our understanding of disease, how we're able to increase preventive health, how we're able to increase the speed and accuracy of diagnosis, and how we're able to decrease the time and burden, and the cost of clinical development, which in turn increases the potential for access to all patients. And so we will use this time to understand where is the state of the art today. We'll grapple with some of the major blockers and bottlenecks that are in the way of true global access to the medicines that are being innovated. And we'll consider what is next for precision medicine in immuno-oncology. So with that, I'll turn it over to each of our panelists for a quick introduction of themselves, the organization or what they do, and how it connects to IO. So let's start with Jeff and work our way around.
Thank you, Bob, and good afternoon, everyone. I'm Jeff Moldrum, Chair of the Department of Hematopoietic Biology and Malignancy and Professor of Stem Cell Transplantation and Cell Therapy at the University of Texas, MD Anderson Cancer Center. I'm the Executive Director of ECLIPSE, which is a moonshot platform that leverages the largest biorepository of longitudinal patient samples and stem cell transplant donor-recipient pairs. And we apply high throughput single cell technologies to identify targets and cognate receptors. And finally, I'm also the scientific director of Orbit, our institutional moonshot platform that develops antibodies from early stage discovery through manufacturing, preclinical IND enabling studies, in early clinical development. And our goal is through commercial opportunities and partnering with industry and pharma to bring new antibody-based therapeutics to cancer patients. And I'm thrilled to take part in this panel. Look forward to a great discussion.
Thank you, Jeff. Jennifer?
Hi, thanks. I'm glad to be here. I'm Jennifer Buell, President and Chief Operating Officer of Agenus. Agenus is an immunology company developing therapies for patients with cancer as well as infectious disease. And we're built to address what we believe to be modern drug discovery needs so that our pace of discovery keeps pace with the exponential growth we're seeing in science. Not only does that require capabilities, but also agility. And with just 250 employees in 4 years, we delivered 15 new discoveries to the clinic, all in development now, some of which are in registrational trials. We're partnering that productivity with our drug discovery and computational platform. We call it our Vision platform. We'll talk more about that, I hope, through the course of the panel. This platform allows us to define predictive signatures that allows us to identify patients most likely to respond to therapy. We published some of our progress in this area. So this year we completed our first marketing application, our submission to the FDA, and we're on track to become a commercial biotech company with a leading PD-1, followed by novel and differentiated Fc-engineered TIGIT bispecifics, anti-CTLA-4, as well as an allogeneic iNKT cell therapy now in clinical trials in patients with cancer, as well as respiratory distress secondary to viral infection, including COVID-19. Thanks for having me.
Thank you, Jennifer. John?
Yeah, hi. Good morning, Bob. Nice to see you again. Thank you for the audience for their interest in the program and their interest in improving patient care. I'm John McAuliffe, Senior Director of Pharma Partnering within the Companion Diagnostic Partnering Team at Roche Diagnostics, and I'm based in Boston. And Bob, for your information, the Red Sox are up by 2 games this morning. I didn't want you to miss that. At Roche, the CDX partnering team transacts various types of projects across the CDX continuum. Those include new critical reagent development, such as antibody clones and ISH probes, It includes new assay development across various technology platforms, including IHC, ISH, PCR, and NGS. And it includes early and late stage clinical trial support, regulatory submissions, and commercialization activities in support of driving market adoption. There's a lot there in what I just mentioned, so thanks again, and I'm looking forward to a good discussion.
Thanks, Jeff. It's early in the season though, so we'll see.
Yeah, all right.
Gerald? Gerald?
Thank you very much for having me participate in this excellent panel. I'm happy to be with all of the viewers at the conference. I am Gerald Batist. I'm the director of the Siegel Cancer Center at the Jewish General Hospital, McGill University up here in Montreal, Canada. And I'm also the chief medical officer of Exactis Innovation, which is a federally funded Canadian National Center of Excellence in Precision Medicine. That is a consortium of 15 cancer centers across the country that has put together pathology departments through to clinical departments, of course, with molecular profiling and at its heart, a registry of patients that are molecularly profiled and ready to go. So we're very focused on the translational research components of a lot of the science around IOs and precision medicine, and we'll have a chance to talk about that as we get into the discussion.
Great, thank you. Niranjan.
Yeah, hi everyone. Delighted to be here. Nice to put faces to names. We're the new kid on the block here. I'm Niranjan Sardesai, President, CEO, and founder of Geneos Therapeutics. We are a venture-backed clinical stage immuno-oncology company focusing on developing products based on our exquisitely personalized immunotherapies for cancer. So these are based on our proprietary platform, GT-EPIC platform, and we're targeting cancer neoantigens using a personalized cancer vaccine approach. Our lead program is in phase 2 clinical development for treating patients with hepatocellular cancer. This is second-line therapy in combination with PD-1 pembrolizumab. And there's perhaps 3 key differentiating features for our approach. The first is that we target all of the targetable neoantigens in a patient. The second is we have the ability to drive CD8 T cells in addition to CD4s. And the third is the needle-to-needle turnaround time. We believe that cancer patients progress, and so we need to be quick about delivering these personalized treatments to the patients. And this also enables optimal designs of clinical trials as well as treatment regimens. So I look forward to the panel and look forward to discussing further. Thank you.
Thank you. And Howard.
Yes, this is Howard Fingert. Thank you for inviting me. And while I'm an industry representative and consultant and serve as advisor to biopharma, academic, and government organizations, I'd like to add that the views presented here, where I've asked to comment, or represent really mine alone. That being said, I'm privileged to have a multi-year appointment on what's called the National Cancer Advisory Board, and has oversight and advisory roles for industry partnerships and resources that are all really designed to support the next generation and R&D ecosystems. And in this role of industry representative, I certainly would welcome hearing from this audience about pitfalls and mitigation strategies, constructive ideas that can improve the current and emerging NCI-sponsored programs. Again, thank you.
Thank you, Howard. So yeah, quite a breadth of experience we have on the panel. So with that, let's dive in and let's start at the beginning with biology and drug discovery. And Jeff, if we can begin with you and your work, tell us how you're developing new understandings in the field of T-cell immunity and how your work directs us towards novel antigen-specific immunotherapy formats.
Thank you, Bob. Well, I think, so to begin, a major challenge to developing antigen-specific antibody and receptor-based therapeutics is really, it's the limited number of antigens that are uniquely expressed on the cell surface of malignant cells and not on normal tissues. And antibodies against such conventional integral membrane proteins is often limited by on-target, off-tumor toxicities, which also culminates in limited efficacy. We've leveraged our ECLIPSE platform to study the allogeneic stem cell transplant setting and to study the coevolution of leukemia and immunity post-stem cell transplant. The curative effect of transplant is mediated by donor T cells, the so-called graft versus leukemia effect. And it's an ideal setting to identify and validate targets and cognate T cell receptors because of the efficacy of GVL. It's more easily studied because of the simultaneously accessible tumor and immune cells in blood and bone marrow. And because we're transferring an immune system that is not tolerized to the leukemia. So by studying the immunopeptidome, we unlock the ability to target cytoplasmic proteins, which broadens the available target space and can identify peptide MHC targets that are unique to malignant cells, such as neoantigen, structural variants, and others. And moreover, our approach, I think, has been enabled by several advances in the field, really, of T-cell immunity over the past 3 to 5 years for peptide MHC target discovery combined with sequencing of cognate TCRs, T-cell receptors and functional T cells. And it's been brought about and enabled by contributions from several research groups in functional single cell, functional and genomic methods, the ability of high throughput instrumentation and optomicrofluidics for manipulating cells, advances in mass spec, and lastly, computational methodologies to really integrate the immunopeptidome analyses with single cell omics and functional assays. And we have the advantage of really having a large repository of longitudinal donor recipient samples collected from nearly 25 years for including 45,000 donor recipient pairs that are fully annotated with clinical outcomes, standard laboratory and pathology covariates, and then by using clinical outcomes to help to distinguish relevant targets. And it provides an additional level of validation and de-risking of targets and receptors. And then finally, we've developed an approach to developing T-cell receptor-like antibodies that target peptide MHC, and we've brought a first-in-class TCR-like antibody called 8F4, which targets the PR1 HLA-A2 myeloid leukemia-associated antigen to the clinic in the first-in-human phase 1 study. So we're very happy to have been able to do that. Currently still in dose escalation phase with so far good safety and on-target effects, but no off-target effects. So this is the first study to really establish that one can develop a safe TCR-like antibody that has some activity. So we've also brought 2 additional antibodies to the IND stage following this similar approach. And so we're hopeful that these new approaches can enable additional target discovery, potential therapeutics for heme cancers, hematologic cancers, and also for solid tumors.
Wow, thanks. And what in terms of getting to solid tumors, what is the bottleneck that in 30 seconds or so, if you can describe that?
30 seconds. I think there are a few bottlenecks. One is, to have an available material from tumors and metastases collected in a longitudinal way to take a kind of a similar approach. But also when you're thinking about developing antibody therapies there, it's the penetration into the tumor, into a hypoxic environment, overcoming that. And also if you're developing these SCFVs for CAR-T, CAR-NK, and CAR macrophage kind of approaches, we still have the challenge of trafficking of the cells.
Right. Thanks, Jeff. Well, Jennifer, maybe that's a nice lead in actually to how your company is leveraging opportunities in functional genomics and computational approaches for immuno-oncology. And if you could describe how your Agenus is going about that, that would be fantastic.
Thanks very much, Bob, and very nice work, Jeff. Congratulations on that. And I think similarly, we've had about 26 years of data ascertainment, which is important. I think that the bottom line here is the criticality of data and our ability to have tools that can make sense of that data, of course, as we continue to evolve our knowledge in this space. For us, the identification of novel targets, the determination of biologic signatures designed to predict response in patients, functional genomics, computational approaches have been particularly useful to us in the evaluation of immune disease or immune tumor relationships right now in patients from the data that we're seeing. So as a field, of course, we've observed, thousands of genome-wide associations of disease-linked polymorphisms. We've also identified a smaller group of those which may actually be implicated in disease pathology or response to immune therapy, and elucidating the causative gene expression and functional changes here really important, and has been previously elusive, and I think in part because of our limitations and the ability to actually compute some of these large data sets. So functional genomics has allowed us to deconvolute the link between genotype and phenotype by making use of very large omic, including some of the single cell sequencing data sets that we've been generating from our clinical trials now over the lifespan, from testing cancer vaccines now to a robust data set of immune modulating antibodies, monospecifics, bispecifics, and now cell therapies as well. So these omic data sets, the next generation gene and epigenomic editing tools to perturb genes of interest and help us to develop therapeutics designed to modulate at the interface of these diseases. And that's what we're really focused on doing so. So we partnered these associations, these findings from these data sets with our therapeutics, and we've embedded our functional genomics process from identification of targets, genes, proteins, through the optimization of leads, these lead compounds designed to modulate those targets through the platform that we're calling Vision. So what is Vision? It's our virtual drug discovery system. It actually recapitulates a patient's tumor microenvironment. It does so in a really profound way, in which we can look at the immune tumor interaction. And we employ artificial intelligence and machine learning that appears, these predictive algorithms to explore a much broader range of the drug biology interactions here. In this, our tools, of course, are designed to evaluate interactions or identify new discoveries beyond any of the human capability that we've been able to do. So we're generating data. We're also providing feedback in real time to enable better drug discovery decision-making. And so we employ Vision, at the idea or the ideation phase to generate, ideal immune therapies in less time. And we're quickly defining sort of why and how these immune therapies are working by characterizing the full life cycle of a T cell in various states and interfacing with the tumor or not. And we're looking at points of intervention, and we envision this Vision platform enables us the quicker validation of these drug targets. It also allows us a really fast way of optimizing the molecular design. Do we use a monospecific? Do we sequence two monospecifics? Do we employ a bispecific approach? Do we engineer that approach? Those are the questions that the platform allows us to actually address. I think in general, we've published on the output of some of these data at a few major medical conferences, including the identification. Some of the most exciting data was a signature that we had identified that enables a prediction of response to an anti-PD-1 therapy in one of our lead molecules, and more than 85%, with more than 85% predictive accuracy in patients with cervical cancer or melanoma. So we're looking forward to what that can do for us. And it's also helped give rise to the pipeline that we have that particularly are differentiating molecules. We have a differentiated PD-1 quite active in patients with positive tumors as well as PD-L1 negative tumors, and FC engineered anti-CTLA-4 generating some really pronounced data in the clinic in patients unresponsive to therapies, first generation CTLA-4s or other immune therapies. So this is how we're integrating effectively these tools into a drug discovery approach, and then we're using that same platform to determine if these therapeutics can allow us to intervene and where is the best point of intervention to modulate the disease process.
Fantastic. Wow, it's very, very impressive. And I'd like to turn over to Howard actually for some follow-on thoughts to what we've heard from Jeff and Jennifer, focusing on biology and drug discovery.
Well, the comments were really appreciated about goals to widen the target space and really past what's been done before, including what Jeff mentioned, the cell surface membranes into cytoplasmic proteins and neoantigens. And but on a practical clinical side, coming in at this as being a translational clinical protocol researcher myself, I realized that the benefit risk that Jeff also mentioned can be a practical challenge going forward. And I do think that from my perspective, the next generation will really be better informed on benefit risk and success in the clinic by the use of biomarkers that are also advancing at rapid pace. And that's why I'm so glad that that John is on this panel as well from Roche. The other things advancing in very practical matters at Jeff's institution, MD Anderson. I want to give a shout out to some of the major initiatives to help with getting those biopsies that Jennifer needs to understand and interrogate what's going on. Alda Tam, an interventional radiologist, developed a very, what appears to be a very successful, innovative approach to having a much higher biopsy rate than I've seen before, and is now being further validated across the National Cancer Institute. And the Biden Moonshot Initiative that Jeff mentioned, that he's part of, the PACT, that stands for Partnership for Accelerating Cancer Therapy. That's also moving very rapidly now, producing transparent publications that are validating biomarkers that are relevant both not only for efficacy and targeting, but also safety, and therefore the overall benefit risk and successful registration and adoption of some of these new platforms that we're discussing.
Thanks, Howard. Well, that's a great lead-in, actually. So let's move from biology and discovery, and as a lead-in, let's move into patient selection, biomarkers, and diagnostics. So, John, let me turn it over to you to discuss how does Roche Diagnostics support personalized medicine from a diagnostics perspective, and specifically, what are some of the considerations that a biopharmaceutical company has to determine and when?
Big question. Maybe I'll try to break it down a little bit. I think the best way to describe our approach to personalized medicine is collaboration. We need to work very closely with our clients, not just taking their order, but trying to figure out exactly what they need to understand what their project entails and to find solutions that are going to work for them. We need to know, is it early stage, is it late stage? Are we talking a prototype assay, or are we talking a design control assay? What's the clinical strategy? What's the intended use? What's the indication, geographies, timeline? All of that is important for us to determine a solution. We need to determine, are there material needs? Do we have the antibody raw material? Do we need to do licensing? Is there manufacturing involved? There is a lot to the development of a CDX, is a part of the personalized medicine approach. Another important part of it is, can we leverage any existing work that's already been done? Part of our business model is to be non-exclusive. That means any new partner may come in, we may already have some work that can be applied to their project. So that's an important thing. The co-development of the diagnostic and therapeutic is very intensive on the registrational regulatory front. That takes a deep level of collaboration that must be considered. And I guess a few points for the audience is that, keep in mind we're non-exclusive. So, the business model remains that the majority of developing the products on the diagnostic side are still underwritten by the therapeutic companies. So one of the take home I would suggest to the audience is start communication early with your diagnostic partners and keep those communication channels open.
And John, from a timing perspective, when you say early, what's the optimal timeframe for that?
Our CDX programs are usually time bound by the clinical development program of the drug. But there's scoping and all the activities that are required in advance. So I would say, at least 12 months before projects start, if you can do that. If you're even considering a companion diagnostic, I would just recommend reaching out to your diagnostic partner as soon as possible. Still, as much progress as we made in this space, too often we come in late and then have to do bridging studies and, perform additional activities that wouldn't be required if we were included earlier.
Do those considerations matter, John, if it's sequencing versus IHC versus other formats? Or?
Yeah, they do apply. I would say anything outside of IHC requires maybe a little more time. Sequencing in particular, pretty new technology that we're talking about in the form of a distributed kit. Lots of things to consider. So yes, if that's your solution, please come to us early.
Cool, thanks, John. Gerald, from your perspective and the work you do at Exactis, how are you and Exactis delivering to drug developers access to the networks and the ecosystems and the registries associated to enable patient selection and molecular profiling efforts?
Well, thank you. As mentioned, we have a consortium of 15 cancer centers across Canada with a very specific contract that not only touches the clinicians, but the pathology departments that have the tissue, the ethics committees, the administration, and all of those. So we understand that clinical research is much more complex if you want to have access to the tissue as described, as well as to the patients. And I think we're profiting from a spectacular expertise that exists in each of our sites. So there are scientists that are cutting edge and bringing novel profiling domains, because we're very interested in multidimensional profiling and incorporating them into a composite kind of profiling. It's an interesting time because there's a almost complete alignment to a degree I don't think has been seen before between the scientific discovery community that's interested in understanding subpopulations of tumors and their molecular profiles, just to understand their biology, and industry that actually needs to find these subgroups and find the drivers so that they can develop drugs. And of course, we're in a period of time when Mr. and Mrs. Walking down the street are talking about using words like efficacy and toxicity and phase 3 trials with regard to the COVID vaccine. And so it's kind of a very exciting time for clinical research. In Canada, we have a very progressive Health Canada regulating organization, and their watchwords seem to have recently moved from protecting the patients, which tend to kind of slow things down, to innovation and access. And therefore it's understood that since we in Canada make use of access to clinical research to access promising agents for patients, it implies an understanding that there's a central role for clinical research in cancer care. And this, I think, is part of our work in achieving this position for all of our cancer clinics. We're also watching the emergence of a still not quite defined era of what we're all calling real world research. We have a number of champions in that area. Exactis is developing this program and experts. One major aim is to start to convert and transform the various special access programs and compassionate access programs into more valuable real world evidence type trials. And that will give some value to them, both for understanding drugs, but also developing biomarkers. As noted, the whole issue of John was talking about the time and the lead time you need in order to develop companion diagnostics, but we all understand that there's a post-approval issue of laboratory-derived tests as soon as new drugs hit the market or hit the clinics, and particularly people interested in the science of, for example, resistance, start to go to work. New diagnostics, new biomarkers are developed that may even lapse the companion diagnostics that went through the regulatory process. I'll give you an example of some work that we're doing now, developing mass spec-based assays for PD-1, PD-L1, and related proteins in the checkpoint inhibitory pathways that may, as a composite, provide a signature that's highly predictive. We have a range of studies with important translational research components, but of course the time is limited. I'll just mention that we also use clinical research as an opportunity to discover, for discovery, for example. We're we have a major program looking at the toxicities related to IOs and efforts that we're making to disconnect the efficacy of toxicity, the efficacy from toxicity in IOs. And some of our folks have recently published a review in Nature Reviews in Cancer on this topic. And solving this problem, of course, is critical to rapidly expanding IOs. And also we're looking for early signals of tumor-specific T-cell activation as a novel cancer screening approach in the general population. So Exactis offers access to patients, scientific and clinical expertise, databases, and practical solutions to turn these into value for drug development, biomarker discovery, or biomarker validation.
Oh, thank you. Thank you. And we'll try to get that citation of the publication out to the audience as best we can.
Thank you.
Howard, I'd be interested in your perspectives on public-private cooperations and factors in terms of optimizing clinical trial design from your experiences.
Well, one thing that Gerald didn't mention that I've had some really strong positive experience, personal experience within running clinical trials in Canada is the quality of the data. Because, getting the enrollment in is one thing, but having substantial evidence and auditing that is really well done, and where you can use the data for filing internationally is also an important factor there, Jerry. But from a technology perspective, Roche offers global scale, in my view, to manage an extensive group of single biomarkers that we heard. But I think their goals are also in line with the theme of this whole program about what's going to happen next generation. I know they've had stated goals, saying about do now what patients need next. Now, from my perspective, that's also do now what collaborators and collaborating biotech sponsors need next. And it really exemplifies the kind of forward-thinking, next-generation thinking to provide a broad and deep technical expertise and service programs and training for end users and information systems. It's not just a little machine that somebody puts in his PATH department. I think all these are critical for BLA NDA filing, but also sustained proficiencies to expand the next generation applications of biomarker platforms and the next generation then of precision medicines. Thank you.
Thanks, Howard. So with that, let's move to the next phase of the value chain, and let's talk about now the actual therapeutics, the molecules themselves, and the engineering of the molecules. Niranjan, how is Geneos designing the medicines that it's working on to activate and target tumor-specific neoantigens?
Yeah, thanks, Bob. It's remarkable, isn't it, that John and the other biomarker and technology companies have made our lives so much easier. It seems like technology is no longer the rate-limiting step. It's what do you do with that information and how do you translate that into biological solutions. That's where a lot of us are sort of dealing with. How do you apply that information? So like many of our contemporaries in the personalized vaccine space, we start out with a tumor biopsy sample. We do the whole exome sequencing. We do transcriptome analysis. We identify what we call targetable neoantigens. So these are the somatic changes between the tumor and the normal within each patient. We are using a DNA approach, so we use plasmid DNA, optimized plasmid DNA to encode these neoantigens, and then we manufacture the product. We create a seed material, manufacture the product under GMP, send it back to the clinical site for administering to the patients. But what sort of sets us apart is maybe there's a couple of distinctions on what the innovations we are putting in place to drive better immune responses and activation. The first is by using an intracellular antigen expression processing and presentation approach. We are looking at intracellular loading of MHC class 1 molecules to try to drive a stronger CD8 T cell response. In combination with our antigen-specific plasmids, we're also encoding plasmid-encoded cytokine IL-12. So IL-12 and the antigens are co-expressed at the injection site, to potentiate Th1 responses. And by doing localized delivery, we're not dealing with systemic issues and associated with, say, IL-12 or other cytokine delivery. It's truly localized activation of antigen-specific T cells. By doing it in a vaccine approach, we're not worried about sort of the intratumoral tumor immunosuppressive microenvironments. We're sort of activating new T cells against these target antigens. But the other piece that we have also factored in is we want to target all of the patient's targetable neoantigens. And our thinking is pretty simple. Our thinking is that we want to let nature select the immunogenic and, more crucially, the efficacy driving neoantigens. So the human immune system is capable of addressing a large number of antigens simultaneously. You know, we're being assaulted by pathogens, hundreds of thousands of antigens every day. So we know the immune system can address a larger repertoire of antigens, and we want to drive that in the context of neoantigen delivery. That raises a second question, and that is around issues of tumor immune escape, oligopolyclonality of tumors. And so we want to, by targeting multiple tumor sections, but we can get to a larger repertoire of neoantigens that we target. But we also looked at the platform in a more holistic sense. You know, it's not sufficient to drive efficacy if you can't get the product to the patients on time. And we're also not doing clinical development in a vacuum. So we are very conscious of this turnaround time. So to give you an example, in HCC, PD-1 pembrolizumab is indicated in second-line treatment. And so we want to make sure that the patient gets their personalized vaccine at the same time that they initiate pembrolizumab delivery, PD-1 delivery. Otherwise, you're forever chasing your tail in terms of are the clinical effects you're seeing, are they because of PD-1 alone, or are they because of the vaccine? So we've looked at our program, seeing what the clinical landscape looks like, where can we make the greatest difference? But the key differentiators here being, being able to get patient-specific products to the patient on time with a larger neoantigenic load.
Great, thank you. Jennifer, I'm curious, you touched on a little bit about the opportunities that Agenus is pursuing in the field of cell therapies, neoantigens, cancer vaccines. Can you talk about those in a little bit more detail? Oh, you're on mute.
Thank you. We had spoken quite a bit about our vaccine platforms previously, and it was based on an autologous. We would isolate the tumor, generate the vaccine, deliver it to patients. And we've actually then engineered that now to a fully synthetic off-the-shelf approach and demonstrating safety and immunogenicity of that vaccine. And how we've approached our portfolio to start, and it's exemplified in a couple of ways. One is the evolution of our vaccines and looking to decrease that time, that biopsy to needle time. It's really critical to get the delivery of that vaccine to patients as quickly as practical. We've been able to do that now in just a couple of weeks, inclusive of our quality testing, which gives us a lot more flexibility for getting the vaccines and combining those vaccines with antibodies. Gerald mentioned decoupling safety and efficacy of checkpoint modulating antibodies. In some respects, what we're trying to do is engineer this in. So decoupling safety and efficacy by engineering more robust molecules that can deliver enhanced immunologic activity without the toxicities. And an example of that then will be our FC engineered CTLA-4. It's AGEN 1181. It's eliminated we effectively engineered the FC region of the antibody. And so that has eliminated complement-mediated toxicities. We don't see any neuroendocrinopathies with the molecule, which have been observed with prior early versions of CTLA-4 like Yervoy. We also see improved immunogenicity, and we can broaden the population of patients who can respond, those who have genetic polymorphism in their CD16 allele. So that's another approach in which we're taking our platforms and using and engineering tools to address biologic problems. Now we move to cell therapies and how we're doing the same thing. What we've been able to address now is we're leveraging allogeneic invariant natural killer T cells, unmodified, and then in their engineered form in the clinic. And we're in the clinic currently. And what has happened is that these cells actually they're powerful subtype of T cells. It also includes features of natural killer cells, and they're conserved, highly conserved. They're important in infectious disease and cancer. They've got an invariant TCR directing them to bind to a dedicated lipid ligand upregulated in cancer and infectious disease. While iNKTs are prevalent in everyone, they are exceedingly rare, making up just a fraction of white blood cells. So again, here's a biology opportunity that has an engineering problem in which no one has been able to scale these cells to make them commercially accessible, feasible, affordable, et cetera. We've been able to do so. So we've solved the engineering problem, and we're able now to scale iNKTs from a single donor into exponential levels now. We've been able to generate thousands of doses from single patients and are able to deliver these cells to patients with cancer as well as patients with infectious diseases. So that's when we talk about the engineering of therapeutics, we've been looking at biology problems that we can solve with engineering solutions that allow us to make our therapeutics accessible, whether they're vaccines, cell therapies, or antibodies that actually have the capability to build on the knowledge that has been generated from earlier molecules and improve on those, improving efficacy and also improving tolerability of these molecules.
Thanks, Jennifer. Well, looking at the news flow from the last couple of days, iNKT cells is clearly in the in the forefront of investors' minds. So Jeff, turning it over to you, you had talked a little bit before about targeting using antibodies to target the peptide MHC complex. Why now, and how today versus what the attempts were before? What's different?
Yeah, this has actually been tried by many groups over at least 2 to 3 decades to develop something that can behave as a TCR mimic or TCR-like antibody, I'd prefer to call them. But we've developed a novel screening funnel, both in positive and negative selection, using a variety of peptides that can actually guide the engineering and the selection for this novel class of antibody. And so I use 8F4 as our most recent example because it's the first one in the clinic as a first in class. And so we've been able to engineer and select these with specificity, with great specificity. And there's a sweet spot for affinity for these kind of agents that I think has been neglected before. Traditional antibody development, you want to have sub-nanomolar affinity ranges for target engagement typically, whereas that's really not something you want in this kind of reagent. And in particular, you have to think like a T-cell immunologist really to build these kind of antibodies. But we can reduce then the potential toxicity and off-target effects, which has really allowed us to accelerate hit to lead and lead out processes for these unique antibodies. And then we've also been able to do some SCFD engineering to engage multiple targets with multimeric structures by tri-specifics, for example. So we can engage more than one of these targets simultaneously on the cell surface. And then we've also been able to build bispecifics that can engage additional mechanisms of action. So for instance, traditional anti-CD3 for T-cell engagement or enhancing phagocytosis. And we've modified the FC region to construct enhanced ADCP and ADCC as well. So it depends on really the biology of the target and in terms of how you want to pair that with the mechanism of action of these antibodies. But because they're unique enough for the tumor, it really allows us to bring in the most potent MOAs to bring to bear on the tumor. And we've done that through other mechanisms too. So for instance, we published on SCFDs from 8F4 in a CAR format, in a CAR T format, which has, of course, you can't compare the potency of that to an antibody directly, but in 8F4 resistant leukemias that we have engineered in the lab, the CAR T approach seems to be be able to overcome that. And then I think finally, critical to really developing these antibodies, and I think this can apply to many antibodies really, is really to have companion deep biology correlates. And I think that we're, and you're hearing that here from other members of the panel as well, who have clear expertise in this field. But we're in an academic setting, and we have leveraged this to develop PDX models from pre and post-treatment samples from each of the patients treated with ADA4 and other antibodies in our development pathway. And it allows us to study the MOA, the mechanism of action in detail, to look at susceptibility pathways, resistance mechanisms, and to test combination treatments. And it also allows us to confirm when we see responses in patients that we've eliminated leukemia stem cells by performing serial transfer studies. So it has really helped us to guide antibody development and even modify clinical protocol to reach our endpoints quicker.
Right. So it's multiple drivers, sounds like.
Yeah.
Okay. Howard, I'd like to turn over to you for some thoughts on the modalities.
I'd rather reserve my thoughts until after the next speakers, because I think it would be sort of relevant to both subtopics.
Okay, perfect. And so what we'll do in the interest of time is kind of merge a couple of topics together: commercialization and access, and leveraging post-approval clinical research and real-world evidence. So Niranjan, when we think about commercialization and access, how do we balance the tension of ever more personalized treatments, all the way to N of 1 trials in some instances, and enabling breadth of access and cost efficiency, et cetera, for a global product?
Yeah, I mean, great point, Bob. I think the sort of the historical perspective on personalization is that these one-size-fits-all therapies don't seem to apply to all patients, and in the case of cancer, many treatments don't even work in, they work in less than half of the patients that they're administered to. So it's sort of the elimination of inefficiencies is the big argument made. And there's been a lot of progress by using biomarker-guided stratification to make these therapies more and more targeted to improve the response rates. And in that same context, personalized treatments, so maybe N of 1, treatments made specifically for that patient are sound very attractive on paper, but the challenge, of course, becomes cost of manufacturing product. If you don't get a handle on it, and turnaround times for getting that product for that patient isn't brought under control, then clinical efficacy alone may not be sufficient to drive a good commercial prospect for that product. So we're sort of looking at this in 2 ways. One is that platform approaches are going to be key. So even though each patient gets their own personalized product, the product is made the same way for every patient. And then, so that's one key criteria. The second is, the increased levels of integration, process automation, and also process validation. Everything driving towards reducing the time component on these turnaround times, and time is money in more than one way. But so all of that sort of drives towards making these increased personalized products relevant in a clinical setting. But ultimately, you still have to deliver a clinical efficacy for the products to be accepted. I just want to take one other segue. Jeff, the work you're doing is phenomenal. What we are finding in our platform approach is that by being able to deliver an increased neoantigenic payload and driving CD8 T cell responses against these multitude of antigens, we're looking at this also as a discovery platform to drive new TCRs and BCRs against these antigens. And so we're looking at about 40 neos in a patient. And so quickly we're developing a large human validated database of PCR approaches that are target. And because these are done in vivo, we know that those peptides are being expressed and presented on MHC class I molecules in these patients. And so the next step becomes, how do you now translate all of that into a TIL or a TCR approach?
Yeah, thanks. Obviously challenging thoughts for us to all consider, which leads actually to a diagnostic question, John. So as the world is moving towards ever more precise patient selections. How does Roche think about, these trials designed for individualized approaches as a business? And along that same line, a secondary question is, as we move towards requiring various solutions for patients from a diagnostics perspective, how does integration work across diagnostic solutions?
Well, I think the era of maybe a single biomarker and an effective targeted therapy, maybe those days are coming to an end. I don't know. I think certainly, the patient populations are going to be more and more select. I kind of view that as multiple biomarkers being applied at once to get the real population that responds to the therapy, perhaps. I'm hearing about the different modalities, and as I'm hearing this, I'm thinking, well, the companion diagnostic business ought to continue far into the future. There's a lot of opportunities here, and I hope that's the case. But just so the audience knows, we have a whole menu of immuno-oncology assays that are at least at robust prototype assay status already. Those include PD-L1 assays for MMR, LAG3, TIGIT, CD8, TIM3, and we have MRD assays as well. So keep that in mind. But I think we're headed toward multiplexing, whether that's 2, 3, maybe 5, maybe even more. I think that's probably where it's going to go. It will be interesting to see how that all shakes out. How much more efficacy will the drug have to show in order to prove, its value and being reimbursed? I don't know. I don't think that the diagnostic will be a big barrier for reimbursement going forward. IHC technology generally is pretty affordable, so long as it's reimbursed properly, that'd be fine. But another slight plug here, we're active in our multiplex technology development. By my count this morning, we already have 14 multiplex assays that include PD-L1 and other markers. So, anybody who's out there and interested in pursuing these hypotheses, please reach out. We're available for that.
And John, how does Roche think about integration of different solutions?
Yeah, that's a good one. I'm not hands-on with it, but we do have a product called Navify, which is in the clinics. And I see that as our short-term solution to bring in huge data sets. I mean, think about where we are today. We have omics and IHC and radiology. We just have massive amounts of information. So bringing that all together in a easy-to-use system that is a bit of its own holy grail. So I think that's our venture into that. But we also at Roche are now focused on the diagnostic side into two major groups. One is Roche Information Solutions and Roche Diagnostic Solutions. So we take it very seriously, the idea that information alone is a very important element as we go into the future.
So in other words, the data is not enough. It's also what do you do with the data? What does the data actually inform?
I see it as a combination of the wet lab work and the information and maybe even pattern recognition within the information itself.
Yeah, cool. And Gerald, you had touched upon some examples of leveraging post-approval clinical research, real-world evidence. Can you elaborate on that?
Well, I'll give you a good example, and I think it also speaks to what we've been just talking about now, and that is what is the clinical context in which all of these data are derived, whether they're imaging, pathology imaging, and then the various molecular profiles. Usually we're testing novel therapeutics in patients that have metastatic or advanced disease, and yet we're ultimately wanting to apply them based on some profiling data that might be from the primary tumor. And so, for example, we looked at, we have a lot of experience with doing serial biopsies of metastatic tumors in clinical trials. And we found in liver metastases of colorectal cancers that there is a subset of patients that appear to be immunoreactive that were not there in the primary tumors. And so we may be able to identify some patient populations that would be amenable to some IOs that were not evident looking in the primary tumor. So tumor evolution is a problem that challenges us in identifying patients and where are we deriving this data from? Are these the patients we're ultimately going to treat? Are we using profiling data from a tumor that no longer is the tumor that we're treating in our experimental setting? And so that immunoactive subgroup of CRC patients represents a really good example of a laboratory-derived profile that we're working on that may be useful, I think, for application of some of these therapeutics.
Great. And Howard, I could turn it over to you for some thoughts.
Well, as I understand the Navify, if I'm getting it right, it includes not only the biomarker laboratory results, but also clinical covariates. And I was very impressed with what the Yescarta people did, a publication by Locke that just came out in 2020, where they looked at both laboratory covariates plus clinical covariates and found ways that the safety profile of the T-cell treatments of Yescarta far improved, as well as the efficacy at the same time, by using both these types of parameters to evaluate the population. And I think the data systems that are emerging now, also one from the NCI, from the PACT organization, will help us not only look at single data sets, but also substantiate them, very important, across multiple data sets and multiple populations to add to our confidence in their use in the clinic.
Great. Thank you, Howard. So let's do a very quick round robin. We've got just a couple minutes left. Let's just do a very quick round robin. Let's assume that everything you're working on that we talked about today is the current generation. What is the next generation of technology, in your opinion, that's going to drive progress in the field of precision medicine and IO? Jeff, 20 seconds each. Jeff.
Yeah. So in the antigen-specific space, I think opening up the cytoplasmic proteins to targeting, and then designing ways of combining those with enhancing antibodies, for example, that like CD47, if you're looking at ADCP, and really modeling that out to deliver the best kind of combination therapy.
Thanks. Jennifer?
Absolutely. Combination therapy, I have to repeat that as well, Jeff. I think that's going to be absolutely critical and foundational to our therapies going forward. I also think that engineering, a novel innate cells like iNKTs as T-cell engagers and otherwise will be really formative.
John?
Multiplex technology to enable bispecific, trispecific, et cetera. And I would say integration of omics information, big database, big data working for us. That's what I see the future.
Gerald?
Optimize the stratification of patients so that we're finding the right patient, matching it to the right treatment, not only by virtue of the patient's diagnosis, but where they are in their trajectory, in their cancer trajectory, using multi-omics and drug combinations or therapeutic combinations.
Great. Niranjan?
Well, I have to go back to maybe the first technological challenge, which was, how do you get genomic and exome and transcriptome sequencing data quickly? I know we've exponentially advanced the rapidity of sequencing, but I'd like to see that go from a week to 10 days down to 2 to 3 days or shorter. So if somebody can do that, I think that would transform the way we deal with personalized medicines.
We'll get right on that.
Howard, your thoughts?
Well, I think I've tried to convey my bias here, and that is that, we've got wonderful resources and partners. The FDA itself is coming up with brand new approaches to address rare diseases. The NCI is producing, commitments to transparency and a lot of new data and resources and technological and protocol ways to help expedite the development. And I think the next generation will be able to build more rapidly by leveraging these resources going forward.
Thank you. And I just received a message from Jennifer, immunophenotypic diagnostics. That's where we need to go. So we obviously, we have so much more we talk about. We just touched really on just a fragment of the ideas that we exchanged in the preparations. So the wonderful panel, I want to thank everybody. Unfortunately, we do have limited time, and but thank you to everybody for sharing the stage, and it's an honor to be part of this panel and to help moderate it. But everybody here, you're the content experts, and I really appreciate your help and support in putting this panel together. So thank you, and also thank you to the SACS conference organizers.
Thank you very much. Excellent panel.
Thank you.
Bye-bye.